I don't, for some reason my mouse is not working, so I might, I'll try to guide folks to different parts of the screen. But if there are any questions, please stop me. I know this is not very conducive to do in a Zoom talk, but just raise your hand and I'll try to answer questions, especially if, because this point is not. So thanks to the organizers for having me here. This is a very nice meeting. Started in the pandemic era. It's very nice that it proceeded on Zoom. It's continued to happen on Zoom. It allows folks from many parts of the world to log in, to listen to what's going on. So really, thank you to the organizers for taking the time to host this for the community. I'll talk about some fairly new work in the group. This work relies on ideas from stochastic thermodynamics, although as I talk about it, some of the inherent techniques that we use as stochastic thermodynamics might not be very obvious, but they'll be in the background, and I'll try to highlight them as we go along. It's very new work. It's done by a brilliant postdoc in the group, Cal Floyd, who's also on the call, and this is joint work with Erin Dinner and Arvind Murugan. In the spirit of this being a workshop, in the spirit of this being the last talk of the workshop, and also this being very new work, I'll put a question mark here to really say that this is new stuff. We think we have some answers, but there's a lot that remains to be discovered. This in this broad field, and I'll try to explain what these figures mean and what I mean. Okay, so this what I have as a start is a circuit that we have sort of encountered in some of the previous talks. This circuit is the circuit responsible for taking the genetic material and transcribing it. The horseshoe-like shape that you see is an activator that has to bind on to the genome. The genome is the curved C-like shape you see. The blue blob you see is a polymerase. And when the activator binds to the genome, attaches or interacts with the polymerase can start to build out mRNA, and this way the genetic material is read out. And that's sort of what's going on over here. And this basic process has been decorated with a bunch of rates. There are binding rates, unbinding rates, so on and so forth. I asked you to focus your attention on two particular rates. I have someone in the chat. I don't know if this is a question or not, but if it's a question, you can raise your hand and just ask me. So I have in this case, I ask you to focus your attention on two particular things: the concentration C that you see on the screen, and also this interaction term eta AB. If you were to play with these two parameters, it's not inconceivable that you'll get graphs that look like this, where if the activation interaction term is very small and the concentration of the activator is very small, the mRNA is produced is below some threshold or is not produced at all. And if the concentration C and the activation interactions are very favorable, the mRNA produced is above threshold. We have seen graphs like this, and we can ask very interesting questions about the energetics associated with getting such graphs, how much flux you need, how much dissipation you need, what constraints you have. What I'll try to do is I'll try to ask questions of that sort, but from a slightly different perspective. In particular, I will start to use language that mirrors language you use in neural networks and computer science. I'll talk about inputs and outputs. So I have colored in green the inputs in this graph, the concentration C, the interaction term eta AB, and the outputs are the mRNA concentration. Okay, and the graph I showed you looks like this. Okay, and so the rates in the network are set up such that there is a boundary of some sort, and below this boundary, in the space of inputs, mRNA is below a certain threshold, and above this boundary, mRNA is above threshold. But now that we have this, we can sort of ask a very fairly broad question, which is how flexible or how amenable are networks like this to drawing more interesting boundaries. So, for example, let's say I wanted a boundary that wasn't just something that looked like a straight line, but sort of curved out. I could have boundaries that are more exotic, that actually encompass regions of space altogether. And if I want scenarios of that sort, the question we can ask is, what kinds of networks should we use? What are the fundamental constraints that allow some networks to draw boundaries, some networks to not draw boundaries? How much dissipation does one need? What kinds of fluxes does one need? And that's sort of the questions we'll ask in this talk. This set of questions can be fairly broad, so I'll try to introduce them in some specific context and tell you the answers we have currently worked out. And so just to sort of summarize, the talk will be focused on what kinds of decision boundaries can be drawn by systems that look like this, but are more complex. How sharp can those boundaries be? And how much can these networks actually classify? So we'll try to build up theories that try to answer questions of this sort. And questions of this sort there is a precedence to asking questions of this sort in a very different field. If you take the classic example of classifiers, and here I have this cartoonish version of a classifier. People have asked questions of this sort for neural networks, not biochemical networks. They've asked questions about decision boundaries. How much can you classify? How sharp can you classify? We want to ask this in the context of networks that are biologically inspired, that sort of have a biochemical like nature. And this is not just we think this has the potential to be relevant. This is a schematic of the kind of network that shows up inside in the Golgi apparatus, and it's a highly simplified schematic of the network that shows up inside the Golgi apparatus, when proteins are taken and a glycan coat is constructed on the proteins, and I'm speaking in very simplified terms. And this glycan coat is a sequence of sugars that get expressed. These get read out by enzymes that are highly promiscuous. But somehow, in all this promiscuity, in all these many degrees of freedom, there is evidence to suggest that glycan codes of this sort actually encode classes. So the greens are my inputs. These are the rates of different enzymes that go in and modify the protein. The output is the relative abundance of the different sugars. And it turns out that there's very good evidence that what the system does is it ends up performing what looks like an excellent classifying task. Depending on for certain kinds of inputs, you have one distribution of glycan codes. For different kinds, you have different. And I have represented that schematically in with these blobs: the red group, the green, blue, orange, and red blobs. And we'll come back to these blobs later. So that's sort of what we want to talk about, and we want to understand what kinds of limits exist in these kinds of scenarios. So to do this thing more formally, I'm going to use the kind of notation that we've seen all through this meeting, and I've actually picked up this from work by Jeremy Owens, Todd Gingrich, and Jordan Horowitz. This is very old notation, obviously, but I'm using the choices that they used in this very nice PRX paper. So I'm going to assume that I have pseudo-first order chemical reactions. I have a master equation with rates Wij that are parameterized by energies, barrier heights, and by fluxes Fij that can encode non-equilibrium driving forces such as gradients, ATP concentration, whatever you have. The energies live on the nodes of the Markov state network, and the barrier heights, Bijs, live on the edges, and so do the fluxes. The Fijs also live on the edges of this network. So the question we ask in the context of systems like this is the following. Let's say I pick two inputs. I pick the affinity F along one link. I'm going to call it FA, and the affinity along another link, I'm going to call it FB. I pick those two as my inputs, and as outputs, I pick the node probability on two specific nodes. Okay? I've colored them blue, and I've colored them red. And what I want is some sort of, I want to construct a network, or to train a network, or to learn a network, such that if FA and FB are very small, all the probability gets, or most of the probability gets shunted to blue. The steady state is such that in the steady state, the blue nodes have the highest weightage. And if you are in the other region, then a lot of the probability gets shunted to the red region. I've described that very schematically in a graph like this. Okay, so can we do this? What kinds of constraints exist? How expressive can the network be? Those are the questions we're going to try and ask. And so how we do this, there's actually a lot of stuff that happens under the hood. We use a specific technique called equilibrium propagation that we adapted to biochemical networks to sort of do this kind of a training. But broadly, this involves taking one of these non-equilibrium Markov state networks, choosing a bunch of points and saying that we want some points to have we want some regions of phase space in which the blue thing lights up, and some other regions of phase space in which the red thing lights up. And then we train the network. We sort of try to learn parameters for all the other rates of the network that allow that to happen. And then we test it. We sort of simulate the network at a lot of other different points. And that's what's depicted in the second thing that just showed up. And then we infer the presence of these kinds of boundaries. Okay, so that's what we do. And what I'll do for the next couple of minutes is show you some examples of what actually happens if you try to do it. What we'll see is that there are some interesting features that pop out. And then I'll use the remaining part of my time to show how ideas from stochastic thermodynamics can actually start to place constraints on the kinds of shapes that can actually show up. And they can also place constraints potentially on how many classes you can end up having. That's what we'll spend the last part of the talk on. Okay. All right. So the first thing I'm going to do is a fairly simple thought experiment. Let's say I show the network a bunch of points in which the lower half is all blue, the upper half is all red, and I ask if the network can, if the system can take such an input, get trained, and actually draw a decision boundary in the right way. If this was an in-person talk, I would ask the audience to sort of shout out yes or no. That'll be too much cacophony on Zoom, so I'm going to assume everyone's engaged and interested. But it turns out that it works. So you can actually construct a Markov state network such that if you give the two inputs FA, FB in the manner prescribed in the graph here, if it's in the lower half, you get the probability gets shunted to blue. In the upper half, the probability gets shunted to orange or red in this case. And just to sort of clarify, everything is happening with just one learned network. So all the dots here in the end, in the final training thing, it's one network that's actually acting like a classifier of some sort. Okay. What if I do this? What if I present two clouds of this sort? I have a blue cloud and an orange cloud. It turns out even here, the network ends up learning. You can do this procedure that I mentioned, and it draws out a very interesting boundary. It sort of draws out a flat region, a slope, and then another flat region. It ends up classifying, which is good. And what if I show something like this, where the clouds are a bit bigger? Even here it works out. Even here the network ends up learning. That makes a small amount of errors, but it constructs a biochemical network that in the end can classify all points in the third quadrant as blue, and the other three quadrants are classified as orange. And that's sort of what this point cloud that sort of reflects some of the training things. So this is all great. So it seems like these networks of this sort can be arbitrarily expressive. They can do anything we want. But things start to fail if you place demands that are a bit more challenging. So for example, we can take these demands now. We can ask for can the network sort of learn the first thing here, where there's a square blue and the rest is orange. I really want to ask, does anyone want to say yes or no at this point? I see a few faces. David Sivak says yes. So it does happen. So it turns out that it does learn the square thing really well. What about the next thing? The next thing with the pizza wedge like shape? David Sivak says no, because David is really smart. And the circle, it turns out is a very interesting case. So the answer to this is this ends up working out. This, it sort of tries, but it fails. and this is horrible. So it doesn't work out at all. So this got us thinking, what are the limits of expressivity? What kinds of boundaries you can draw? Can you draw arbitrary squiggles? Can you do you lose things if you want to draw pizza like wedges? What are the fundamental limits for doing all these things? And this is where Cal did all this in the last few weeks, which was super nice. Cal figured out a way to explain how one can set bounds on expressivity. And this goes back to some really interesting work that Esposito and Timur also have in a different paper. We sort of stumbled upon it in a different form in our preprint here, which is this property that if you take the derivative of the steady state of the non-Euclidean steady state of any of these Markov state networks, and you take the partial derivative of that with respect to any of your Fijs, any of your affinities, it turns out that this derivative is constrained to have a fixed sign if all the other parameters are held fixed. So it's either always positive or it's either always negative. The way we proved this is using matrix tree theorem and using some of the beautiful insight that Jordan, Jeremy, and Todd worked out in the previous paper that I referenced. It can also be worked out from work from Massimiliano and Timur in a different manner. They wrote us a very nice note a few days ago. But this property seems to hold. So what does this have to do with the shape of these kinds of curves? Let's do a small amount of math, and there won't be too much more math in this talk. I'll sort of try to keep it simple. That's the last talk. Everyone wants to go home. So let's say I construct this very simple implicit function of FA and FB, which is just the steady state as a function of FA and FB. Theta are all the other parameters in the network that I don't care about. Those are already learned. And I look at where the steady state crosses half. So that's sort of a proxy for this kind of a boundary. And this implicit function can be really messy because you have trees that inform the non-Euclidean steady states. You can have a lot of things. Going on, but we can interrogate something very simple. We can ask, well, what does it take for this curve to actually change sign? Can it actually form a full circle, or is it constrained to just form a quarter circle? And it turns out it is from the relation that I told you, it is constrained to only form a quarter circle, because if you require it to form a full circle, you have to break the all positivity or the all negativity condition that I mentioned in the last slide. And this sets a fundamental bound on how expressive networks of this sort can be. And this works in arbitrary dimensions. If you have two-dimensional input, you can do quarter circles. You can do this kind of a curve. If you have high-dimensional inputs, you do that in high dimensions, and it just works out. So this sets a constraint, and this is the reason why circles were not classified. But anything that was not a circle, even a square, it was able to classify. It was happy to do it. So this is nice, but it's also somewhat disappointing, because we would expect systems of the sort to be much better objects, to be much better classifiers. We hope that biology uses these kinds of ideas because it can actually act like a fairly robust computer. And it turns out that actually does happen if you allow for multiple edges to simultaneously be connected to one variable. And this is not something that is unheard of in biology. So if I have over here a schematic of an E. coli network on the left bottom, where the ligand concentration that the E. coli chemotaxis network senses ends up affecting the whole range of nodes. The same kinds of things happens, for example, with kinases, with the glycan code, with many other processes. And if you do have this kind of a constraint, then it's possible. If you do have this kind of a condition, it's possible to sort of overcome this constraint of a single sign for your partial derivative. And in fact, over there, if go back and we ask ourselves whether situations like this, where you have a full circle, if they can be solved or not, with the if every input is affecting just a single node, this didn't work out. But now we can actually solve this problem. If you allow an input to affect multiple nodes, I think here it was an input affecting 2 or more. I forget what exactly it was, but it ends up solving this problem. So how do we understand this? Is there a mathematical way to understand this? We have a partial answer which I'll show you, but I want to stress that this reveals some interesting biological insight in our view, because a lot of the information processing enzymes in biology, they are promiscuous. They don't act on a single on just a single link and stop. They have functions that couple to multiple processes, and this inherently allows them to draw decision boundaries that are more interesting, that are more exciting, that can sort of encompass, that can do more, that can sort of do better computation defined in this way. So let's try to ask if we can understand what this is, what's happening here, and how we can get around to some picture of how tuning the number of edges that are being tuned by one parameter can give us more flexibility. So let's say I set M to be the number of edges that are being tuned by one parameter, and D is the number of inputs. What I can do as a very, very simple metric is I can compute, for example, the number of times the derivative, this partial derivative, can change sign as the Fij is tuned. We saw that when M is 1, if only one input is allowed to affect things, this was 0. It can't change the sign at all. And so we can ask, does this thing get better? If M increases? If you just have one input, but the input affects different multiple nodes. So if D is equal to 1, but that input can affect multiple edges together, then it turns out we can actually write down a bound, again from the matrix tree theorem. We can show that the number of times the derivative can change sign as a function of f is constrained by 2m minus 1. For m equal to 1, this bound is still an upper limit. So we never get to the point where the derivative changes sign, and that we can prove. We can prove that from the matrix tree theorem. But it turns out that for 2 and 3, if you set m equal to 2 and 3, Cal exhaustively sampled many networks and numerically verified that this actually can show up. So you can actually achieve situations like this. So what you can have is you can have scenarios of the sort where pi k can go up and come down in this manner when m is equal to 2, for example. And this sort of mimics the circle-like construction that's showing up on the left top half of the screen. So if you allow for m to change, there are potentially constraints. There are potentially more number of classes, and we have a very simple metric to sort of anticipate how much that can be. If you go to higher dimension, this is still work in progress. We can work out something called the VC dimension. It's a weak bound. It's not very informative in many cases, but it's a very broad, it's a very loose measure of how expressive or what kinds of shapes you can draw out. This dimension ends up being 2 to the power of m plus 1, 2m plus 1 whole to the power of d. So it suggests that things are more expressive as you add m, and it becomes exponentially more expressive. But this is something we're still working out. We are still trying to get a better handle of this thing. So finally, what determines expressivity? Where is being far from equilibrium important? We can get some sense of that by writing down the steady state probability, again using the matrix tree theorem. Here, my theta are all the other parameters that are being tuned, that are being learned in our training process, and x's are the inputs. So these are the FAs and FBs. And so it turns out we can generally express the steady state in the somewhat cloaked manner. All the tree architecture and all that is contained here, and we can interrogate this a bit. And in the interest of time, I'll just show you what ends up happening. What ends up happening if you interrogate this, the structure of the steady state, is that if you allow for non-equilibrium affinities, Fij's in the network, the number of different ways trees can start to couple together starts to become very high, and the system ends up having the ability to be more expressive. We can use this insight to say something more about things like sharpness, but I'll also make one small point, which is that the steady state has a very interesting property. It actually looks like a cross attention mechanism, with a linear similarity measure an exponential feature map. So I'm almost out of time, so I'll end here. I'll say that one can say similar things about how sharp the decision boundaries can be. We can sort of connect this to the kinds of trees that show up. We can also sort of talk about how well the networks get trained, and how, as they get trained, they actually start to localize probability really well. So we have metrics for that. But I'll end with this sort of thing, with the slide that I threw up. We think it's interesting to ask questions about these biochemical networks, but in a language that's borrowed from different fields. Ask about decision boundaries. We think ideas that underlie a lot of the stochastic thermodynamic progress can help set what kinds of shapes you can draw and how sharp they can be. We also think they can help address more interesting questions of how many modes a network can classify. But that's all work in progress, and that's something we are still thinking about. So I thank you for your time. For the folks in Europe, I'm guessing it's close to 6. So I apologize if I've kept you for much longer, for longer than I should. But I'm happy to take any questions. Thank you.